This is a production of Cornell University.
But what I want to tell you a little bit about is how we're starting to use some of the concepts of evolution and machine learning and combining those together to really figure out and drive functional variant discovery and what we hope will eventually become breeding 4.0. I want to first highlight that the work I'll be telling you about today has been supported by USDA-ARS and the National Science Foundation, but the real thought and efforts behind all of this type of work is coming from an incredible group of collaborators I've had the pleasure of collaborating with over the years. Sarah Miller was helping me count up all of our collaborators over the years, and we've actually collaborated with like 1,200 people now over the last 20 years. And that working across that global community has been such a pleasure to learn from everybody and to make progress in this. And this is some of our current collaborators right now that have helped us shape our thinking. I want to give you all a little feel for where we're driving to, and that comes out of our lab's mission. And so our lab's mission, the very first element is a very straightforward one. What's the genetic basis of natural diversity? Simple to say, hard to figure out. But essentially, how does a change in DNA get converted into a change out in the field, something we can see and work with? With models of those, we can then apply that knowledge to help breed crops for the US and for the world. And so I won't be talking a lot about what we're doing there, but we try to use our understanding at the top part to help people across the world build engineering and IT systems to help breed crops for the world. And then finally, we do all this in the context of what should we discover and what crops should we breed in order to reduce the environmental impact of agriculture. So I've also, in the last couple months, been giving a number of talks down in Washington, D.C. And so frequently I'm asked, what is breeding, okay? And that actually is a harder question than I thought initially, because we all around here think we know what breeding is. But this is the definition I came up with. And essentially, we view this as the design and selection of organisms for human purposes. And so if you start asking, what could we apply breeding to? And if you go through the UN Sustainable Development Goals, they lay out 17 goals that are critical across the globe for the next 20 years. And where could breeding contribute to each one of these? And it turns out that breeding can contribute. I'm not saying it's the dominant element or anything, but it can play a role in 10 out of the 17 Sustainable Development Goals. So the reason that I'm interested in taking quantitative genetics and applying to breeding is because the leverage of what we can do and impact the globe is so great. And so I think if each of us can play a little role in figuring out how we can solve some of these 17 issues, we can have a real impact across the globe.
Is there any reason you don't have number 4 for it?
Quality education. Maybe it's 11 of them, I think. Excellent question. So in terms of the evolution of plant breeding, though, where have we come from? As was mentioned, I have this background in archaeology, so I always think about what breeding is in quantitative genetics from this very long time depth. And so 10,000 years ago, I would say we started out what's called breeding 1.0. We were using phenotypic selection to decide what species should be domesticated. There's 250,000 plant species out there. About 5,000 of them have been domesticated to some degree. And people were really testing out species level diversity and applying selection to some of those species to really drive them into new and different directions. Starting a little over 100 years ago, I think the key element was we started having controlled trials where we controlled the genetics. Out of that came our understanding of quantitative genetics. Experimental design were really the key elements in allowing us to make progress in those statistics rises out of our knowledge of plant breeding and our desire to do plant breeding. And of course, equipment and tools like harvesters really help drive our ability to provide and produce selection effectively. Then a little over almost 20 years ago now, genomic selection arises, where we're essentially using anonymous markers across the genome to really drive selection and produce crops faster and better. Now, for our most sophisticated breeding programs across the globe and major companies, genomic selection is now the norm. It's being applied in all of our major crops in each of those companies. But I think one of the challenges we have across the nation and across the globe is to make sure genomic selection, breeding 3.0, gets applied to those 5,000 other species that have been domesticated, or to all the environment and all of the breeding efforts across the rest of the globe. So although I'll be talking most about breeding 4.0 today, we also do spend a lot of time, and I think our whole community should spend a lot of time making sure that breeding 3.0 gets applied everywhere. So finally, breeding 4.0 is really about designing genomes. And in order to do this, you have to identify the causative nucleotides that are underlying quantitative and complex trait variation. And then we could imagine using editing technology and our understanding of physiology and molecular biology to really drive things forward. And I'll talk about how we're doing some efforts to help us identify those causative nucleotides. So when I think about where variation comes from, I think the first place to kind of think about it is, every generation, 90 new mutations occur in the case of maize. And some of those are adaptive, some of those are deleterious, and most of them are neutral. And fundamentally, the question that our group is trying to ask is, of those 90 mutations that arise every generation, and they of course, are passed on generation after generation for thousands, even millions of years, which fall into each of these different categories. And that's what we're trying to ask and trying to figure that out as easily and fast as we possibly can. And why I care about speed and cost is because there's a lot of genetic variation out there. In the case, a small sample of maize lines, they capture about 80 million different genetic variants across the genome. And we think there's on the order of a few 100,000 functional variants that are really contributing to most of the phenotypic variation out there. The question is, how do we go from the top of that funnel to the bottom of that funnel as cheaply and inexpensively as possible? And so it really is a question of efficiency to move from the top to the bottom. So we've worked on a lot of the different methodologies across here, and I'll talk about a couple of them today with special emphasis on using conservation, expression, and machine learning to get from the top to the bottom. And I want to start out with highlighting that evolution has turned out to probably be the most powerful tool we have for figuring out what are functional variants across the genome. And of course, this shouldn't come as a surprise to anybody. Jablonski 50 or 60 years ago reminded us of this, that nothing in biology makes sense except in the light of evolution. But just to give you kind of a quantitative example, how powerful evolution is at identifying things, let's think about field trials. So a decade or so ago, we did create the maize NAM population. It had 5,000 inbred lines, and through a lot of work, we created them. We grew them out at, for example, one experiment, 8 location, lots of clones at each one. And effectively, we were able to map a 5 kernel per plant difference down to the level of an individual gene. Okay, so that in many regards seems like that's a pretty good mapping accuracy and everything. But how does that compare to evolution? And so, well, so the key thing to realize about evolution is it's really the integration of billions of years of field trials. Every individual is competing. It's either surviving or not, or dying. It's either passing its gametes on. And so in a diverse species, for example, maize, if we sample 1,000 different maize lines, we're capturing about 3 million years of evolutionary time by having a sample of 1,000 lines. Those 1,000 extant lines are the product of populations that had an effective population size of nearly 100,000 individuals. And the key thing is selection can see a difference of 1 kernel out of 250 plants. So in comparison to what we were able to do in a very large field experiment, it's about 1,200 times more sensitive than what we can do in a large field experiment. Because it's effectively, we have the time and evolution to integrate across all those different events. Now, what's the problem with using evolution? The big issue is we don't really know what the conditions were under which selection operated. And so that's the great weakness of using evolution. But we've also gotten in the last couple of decades at getting pretty good at identifying what regions of the genome have been the product of domestication or the targets of domestication. And in those regions, we actually know those are the regions where the rules of selection have changed. Right? When we talk about a domestication locus, what that really is a region in the genome where selection is going in a different direction today under human selection than it was under natural selection. And so in the case of maize, which is probably our most highly domesticated crop out there, there's about 1,000 domestication or improvement loci across the genome, which is about 4% of all the genes out there. So from one point of view, no, we can't be sure what's going on there, we can't use evolution as a guide necessarily to pick out what is the correct direction to take those loci. But what this does suggest is that the other 96% of the genome, essentially the rules that have played for the last few million years are probably the same rules in important directions that are important today. Photosynthesis in maize still operates pretty much by the same rules as it did 1 million years ago, 5 million years ago, and so on. So as we use that knowledge to try to figure out deleterious variants, we've used essentially evolutionary conservation to work down this trail. And so let me explain how we do that. Well, the first thing about, in order to use conservation, we were using it to try to tackle a question about what produces deleterious mutations, and then what produces heterosis. And why we care about deleterious mutations is if, over 100 years ago, geneticists figured out that essentially deleterious mutations are likely the product or what produce heterosis and hybrid vigor. And essentially we complement them and we get out hybrid vigor out of the system. And of course, the whole maize seed industry has grown out of this. And so I think the question of what is the basis of heterosis is pretty much a solved question for the last 100 years. The question that has not been solved is what are the individual nucleotides across the genome that contribute to heterosis? So the basic process we've understood for a century, but what are those individual deleterious mutations? And so a former postdoc in the group, Eli Rogers-Melnick, tackled this by asking the question, let me take the maize genome and line it up across a wide range of other species and identify what regions of the genome are conserved across this wide range of species. And he lined up maize for everything all the way out to Arabidopsis and grapes and so on. And what he found is about 10% of the genome was alignable and constrained. I'd highlight that most of what he's talking about are the genes. These, the intergenic regulatory regions do not align across this range of diversity. It's pretty much just genes that align at this scale. So, but he was able, base pair by base pair, able to ask the question, how conserved is every base out there? And then if you see a genetic polymorphism at a base that's incredibly conserved across this entire phylogeny, the odds that are deleterious are substantially increased. And so the same type of process is also now being used by people in human genetics to identify disease loci and so on. And so Eli applied that to our question here in maize. And we have a very strong prior of what we think should happen with deleterious mutations. We know that the key way to get rid of deleterious mutations is through recombination that breaks things up, and then essentially selection can eliminate them. And so we expect to see a pattern such that in low recombination regions of the genome, we have a buildup of deleterious mutations, and in high recombination regions of the genome, we have fewer. And that's exactly what we saw, is that in low recombination, we had a buildup of deleterious mutations, and in high recombination, we had fewer of them. I do want to highlight, though, that there are still lots of deleterious mutations in high recombination regions of the genome. The genome is about 90 mutations are occurring every generation, and they're all across the genome, pretty much evenly across that genome. And the genes are in the high recombination region. So yes, there's a buildup. The population genetic prediction that we are able to purge deleterious mutations better in high recombination is true, but there are still deleterious mutations everywhere, just because the pressure of mutation is so strong. And so Eli then lined up a lot of those deleterious mutations with a couple different aspects of the genome. And the first one he wanted to test was how did this line up with where people had previously mapped heterosis in maize. And in the early 1990s, Charlie Stuber had identified that the paracentromeric region of chromosome 5 exhibited the highest level of heterosis in maize. And so Eli took his model of deleterious mutations, mapped them across chromosome 5, and that's exactly what he saw. A buildup of deleterious mutations right in the paracentromeric region of the genome, and a far less buildup across the rest of it. The green is the recombination rate, and as you'll see in maize, we have a very low recombination rate all across that paracentromeric region. And so it's very difficult for the genome to purge these deleterious mutations in those regions. But so the pattern in our agreement with heterosis for this analysis made a lot of sense. So as I mentioned, the way we were doing those conservation analyses, we really could only look at essentially genes and pretty much conserved proteins there. And so the second question we wanted to ask is whether or not we were seeing the same type of patterns looking at RNA and expression patterns. Some other analyses we had done that suggested about half of phenotypic variation is the product of expression differences, half of it's from coding differences. And so is the genome able to purge deleterious mutations better in these expression domain, or does it do worse at that? And so this is where Carl Kremling, a former graduate student, worked on this question. And the real element and key for doing this analysis is we needed to collect enough expression data from enough different lines that we could determine what was a rare and aberrant expression pattern. And so what he did is he got robotized 3' RNA-seq profiling up and running, got it down for about $25 a sample, and he was then able to profile nearly 2,000 samples for their RNA levels. And with that large data set, we then essentially had expression measures on tens of millions of different genes and alleles all across this panel. That effectively allowed us to start asking questions about what are rare expression patterns. And the hypothesis he wanted to test was one that Greg Gibson had laid out in human genetics. And essentially the hypothesis is as following, and the test is as following. So we kind of expect low expression, if you bin expression patterns out for each gene, if rare alleles and deleterious alleles are playing an important role, what we expect to see is that essentially low expression will be the product of lots of rare alleles, and high expression will also be the product of lots of rare alleles. Well, intermediate expression will, won't have an enrichment of rare alleles. And so this is the basic pattern you would expect if you think that rare alleles and deleterious alleles are driving extremes in expression. And so what's the alternative hypothesis to this? Well, the alternative is that common alleles actually get stacked up in a way such that the extremes are the product of lots of different common alleles. And so is that likely to be common? Well, across the whole genome, when we see extremes in phenotype like flowering time, it is the product of dozens of genes with essentially relatively common alleles getting stacked up in order to produce those extremes. Of extreme early flowering is the product of lots of common alleles being stacked up. Extreme late flowering is the product, again, of lots of common alleles being stacked up. So at a genome-wide scale, that's the pattern we actually do see. Common alleles in general are what are contributing to a lot of our extremes in phenotype. But at the level of an individual gene, recombination is, of course, very limited. There's a limited amount of recombination in each regulatory region. And so this is a type of area where extremes may be the product, actually, of a buildup of deleterious mutations. And so in Greg Gibson's model, what we would expect to see is essentially a U-shaped plot here. If you were to plot out the average number of rare alleles on the Y, and then on the X, we go from low expression to high expression. We expect to see a U like this, if rare alleles are driving things. And that's exactly what Carl saw. So when he profiled using these different maze lines, essentially low expression, where we see low expression, we see this more than doubling in the number of rare alleles. And we do also see an increase in high expression as the product of an increased number of rare alleles. So both extremes seem to be pushed by rare alleles. The asymmetry between these two suggests that rare and deleterious alleles, it's easier to knock down expression than it is to increase expression. But both directions, we see that extreme on both sides. And so where you really want to be is somewhere here in the middle. That's the sweet spot where where expression is. It's somewhere in the middle. You don't want to be at either of those two extremes. And that basic hypothesis about extremes and the wanting to essentially, the optimal organism is an organism that essentially has balanced expression everywhere is effectively the dosage hypothesis. Jim Birchler has been advocating this for a number of years as the basis of maize heterosis, and it works well with a model with deleterious mutations and so on. But essentially, let me explain the model. Essentially, and if you have balanced expression in all your genes, your protein complexes, your metabolism, everything else is kind of goes as expected. That produces a healthy plant. If you have extremes in expression, your protein complexes don't form correctly, metabolites, you get buildup of the wrong metabolites at the wrong time points, you essentially are wasting energy all over the place if you are not in the center everywhere. And that produces weak and inefficient organisms. And so in order to be optimal, you should be average everywhere. That's is kind of is the key element there. And so that's what the basic model in heterosis works just fine with this type of model, that essentially be average everywhere and you will be highly vigorous. And so what Carl did is he came up with a very simple statistic of just cumulative dysregulation across every gene in the genome. He didn't give any genes greater weight or a different weight from one another. We know that's very naive, but essentially cumulative dysregulation does correlate with seed weight or yield. He also used an approach where he created a statistic based on the deviations in expression in order to try to predict the amount of variance explained in seed weight. And here he was able to find anywhere from 20 to 30% of seed weight is being explained by this dysregulation, these patterns of dysregulation. So we think that maybe about half of phenotypic variation is coming out of processes that involve these dosage aspects of essentially not having balanced dosage at each gene in the genome is producing a lot of the phenotypic variation that is out there. Okay, now I told you about those two different approaches that we were using to get from the top to the bottom, but if we really want to get down to individual nucleotides so that we know, can start talking about editing and so on, how do we get there? Part of the problem with all these deleterious mutations is that they're rare. It's very difficult to map rare things, okay? And we don't want to spend a whole bunch of time taking every rare allele that's out there and increasing its allele frequency in order to map it. And so this is where machine learning comes in, where we're effectively learning the rules of how the genome works so we can spot these rare and deleterious mutations all across the genome. And are starting to think and apply this all across essentially the central dogma and beyond. But what I'll be talking to you about today is how we're using machine learning to model both chromatin structure and expression patterns in maize, and actually in maize and sorghum. And so, why are we talking about machine learning? Well, obviously it's a hot topic. You're hearing about it all the time. You can be photographed, and machine learning models will identify you incredibly quickly. You walk underneath a camera, it knows exactly who you are. And the fact that that can be done, and can be done for us, why shouldn't we apply these incredible tools to understanding molecular biology too? And the key reason machine learning does work, though, is it allows us to process massive amounts of data. It allows us really to generalize rules from one portion of the genome to the rest. It allows us to generalize rules from a reference genotype to all the other genotypes. And finally, if the model is robust enough, it can be used to really determine if a given genetic variant matters. And I think some of the pushback on machine learning has been historically that people, especially in the basic science world, is that it's a black box model, okay? And absolutely, you can treat machine learning entirely as a black box model and not understand the underlying biology that is underlying it. But as I'll show you, we've been doing a lot more work to essentially, once you create the model, try to probe the model and ask the question, what's going on? What did it learn? And now can it teach us something about what was going on there? So I'm going to tell you about a series of models, and these were developed by three great postdocs in the group, Catherine Mejia, Hai Wang, and Jacob Washburn. And the models are fairly straightforward to explain. What we wanted to do was take sequence and just sequence. So no other annotation on the sequence. We don't want to know what methylation status is. We don't want to know what any of the chromatin structure. I just want to take sequence, and from that, be able to predict the following. Is the chromatin open? What transcription factor binds there? Is it a pseudogene? And what is the expression level of the associated gene next to that sequence? And the second class of models is if I have two homologous sequences, can I differentiate the expression of allele A versus allele B and figure out which one has the greater level of expression? So those are the 5 models we've been exploring the last year. And essentially, we're using a couple of different approaches. The first is coming from natural language processing, very similar to when you type something into Google and it's trying to process your language and figure out what you're after. And what we've done here, Catherine's worked on these K-mer language-based approaches. You're breaking a sequence into K-mers, and then, as I'll show you, you're treating it as essentially a bag of words that we're modeling. And then the second class of models are these convolutional neural nets. Historically, they've been used to essentially do image processing, and we're essentially converting sequences into ordered images, and then we're using the CNNs to process them at different scales, and then using neural nets to integrate these process sequences. So a little bit more detail on what the K-mer bag of word type models are. So let's take DNA, and effectively, it could be treated as a bag of words, very short sequences, or the K-mers. We take those, we count them up, and then we can essentially fit them in a regression model to make a prediction. And a bag of words model is no more complicated than that. The vector-based ones, which rely on the ordering of the words, is more complicated and some more powerful. But essentially, the bag of words one is as simple as just counting up K-mers and fitting it to a regression model. Okay. When we talk about the convolutional neural nets, we're essentially taking a sequence, and each base pair is getting re-encoded. And so what you're seeing is essentially we're making a black and white picture where A's are encoded here, C's are encoded there. And then from that, we take convolutions. And what convolutions are doing is that they're looking across that image and trying to highlight various features at certain scales that then can be passed on to the neural nets. And so those convolutions essentially probe the regions, summarize that information there. And then once we have those convolutions with neural nets, we can relate those convolutions to any trait or attribute. In our case, we want to relate that sequence to expression levels. And so a little bit more about how we measure whether or not we've been able to create machine learning models that are of any good or any use. We use what are known as the ROC curves, or receiver operating characteristic curves. And essentially, these are just ways to count how well you're doing in measuring, comparing your trues and false positive rates. And essentially, the way we've set them up for this talk here, our random expectation is along the diagonal. And we expect that to essentially produce an area under the curve, or a ROC curve of about 0.5. If we have a perfect classifier that's able to completely differentiate 2 separate classes with no errors, it'll follow along there, and essentially its area under the curve would be 100%. And so this is one way we measure whether or not we've been able to create a machine learning model that is of value. And so Catherine took this type of model. She was using the ChIP-seq data from the transcription factor Phi4 to train on. And she was able to, using that bag of words model, be able to create a model that had very high levels of accuracy at 0.99, and essentially was almost perfectly classifying where each of those transcription factors were binding all across the genome. And it wasn't just that transcription factor. She used the same ChIP-seq data from NODET1, also very high levels of accuracy. We had MNase open chromatin data that she was able to use, and again, it was able to train very good models there. And then she also was able to classify core promoter sequences with high levels of accuracy. So essentially, And she's been able to now repeat this for about 100 other transcription factors with other collaborators. And effectively, we can now create machine learning models that model where transcription factor is going to bind with high levels of accuracy and be able to essentially start annotating all sorts of other genomes with it, especially within maize. But one of the questions we did want to ask is how good are these models transferable from maize to, say, other species? And so Catherine asked that question. She took her core promoter model, and so it was great in maize. But then she took it to sorghum. It was okay. It's not, the transferability from maize to sorghum wasn't as great as we would have hoped there. Now, we know strategies about how we could make a model that would work across both of them. The key element is we would probably train across both maize and sorghum simultaneously, rather than just training on one species at a time. But some of the models, for example, her not-at-one model for maize, it actually had superb transferability all the way to rice. So that one transferred 65 million years without a problem. So each of these models, some of them have high levels of transferability, some don't, but there are strategies for figuring out and improving the transferability across species. These K-mer models are also really, we talk about the black box aspect, all of that was pretty much black box. You can open up the models and ask, okay, how did the model figure out that this is a core promoter? And so, of course, we know the Tata box should play an important role in defining a core promoter sequence. And so what she's plotted out here is essentially the weight of each K-mer, and there's thousands of K-mers she was looking at, but these are the Tata-like K-mers. And the most important ones for defining a core promoter are listed here. And as you see, they line up right next to where the transcriptional start site is. So essentially, this black box model, the way it learned how transcription starts in maize was finding the Tata box and learning essentially the rules that, of course, obviously everybody's known this for years, but it was able to learn effectively the right rules on how this works. Okay, we've also, though, found that training models can be tricky. Machine learning models, they're incredibly powerful. They can learn and memorize virtually anything. The question is whether or not it's learning what you want it to learn. And this is where the last 6 months or so, we've been really doing a lot more work to make sure we're training on the attributes of the models that we really care about. And it turned out that a lot of our models, especially when we went across lots of different genes, our models were effectively able to memorize the evolutionary history of how every gene is related to every other gene, okay? Which isn't, that's an interesting evolutionary story, but doesn't teach you the rules of how molecular biology works and exactly how a promoter works. We didn't want to just memorize evolution. So what we've done is we've started to think through how can we use evolution as our aid, not something we just memorize. And so we've set up two different ways of setting up our training versus our testing. The first one, Hai Wang and the group set up, was essentially gene family guided training, where you train on one set of gene families. Say you take half the gene families across maize and you train on them, but you hide all the other gene families. But you test if your model is doing very well with a group of gene families the model has never seen before. If you are learning things that are transferable to gene families you've never seen before, then it's likely a good model. It hasn't just memorized recent evolution in order to figure things out. And the second way to do it is to essentially take a couple related species to one another and essentially compare the orthologs against one another in order to do set up the contrast there. And that's what we call the ortholog contrast training model. And I'll go into that. And Jacob Washburn has been leading the effort in those types of analyses. So in that first one, Hai wanted to ask the question, can he train a model to identify what are the pseudogenes across the genome and which are the genes that are highly expressed? And so formally, he's taken the promoter sequence, the UTRs, about 1,500 base pairs up there, 1,500 base pairs from the 3' UTR and beyond the terminator, throwing that into a model, letting the convolutional neural net then predict expression levels. And what he did is he took a data set where we had expression across 66 different tissues, and he took the maximal expression level, and he asked the question, can he differentiate between the genes that are never expressed versus the genes that are expressed, that's the combination of these two, or just the really highly expressed genes. And I'll preempt this next slide. We're a lot better at differentiating between this group versus that group than differentiating between here and all of these. But the model is still quite good. And essentially, when you try to differentiate between off versus high, he has about an 80% accuracy there. And if you just use either the promoter or the terminator sequence, and if you combine the two of them together, we get a substantial boost. So essentially, signal for whether or not the gene is on or high is coming from both the 5' and 3' end of the gene. And we can create a pretty good model, classify each of those. The second question that we were interested in is which allele is more highly expressed. Here we're taking two homologous sequences. And from a person with a quantitative genetics background, this is really kind of the fundamental question I have of molecular biology. I want to know a given variant, does it change, does it make something go up? Does it make it go down? Or does it have no effect? Those are the once I get down to the right side of those, then I can throw it into a more complex model of quantitative genetics and parameterize everything. So this is the style of model I really want to use in order to parameterize the entire genome. And the way Jacob has gone about this is essentially reframing it using these orthologs. And I mentioned we're essentially comparing the orthologs in two different species, training on them, and then testing on a different set there. So instead of just having one sequence and making a prediction of phenotype, we're taking two sequences and comparing them. And what we found is that rather than actually predicting exactly the difference in expression, we're most interested in really predicting whether or not it went up or it went down. And I'll also highlight that we found out that there's a lot of genes, especially when we have to compare between species, that very subtle differences in expression, we're not very good at predicting whether or not a gene went up or down slightly between maize or sorghum. But if we take a look at the top 20 or 20% of the genes that really moved up and down in expression between maize and sorghum orthologs, then we have very good prediction accuracy. Okay, so, and some of this is maybe our ability to be able to really tell whether or not something went up or down isn't great if the expression differences are really slight. So Jacob took this model, very similar to what Hai, or exactly the same way that Hai broke up the sequences here, but he's essentially taking two genes in the model and then letting the convolutional neural net pick out which ortholog is more highly expressed. And so the pattern here was somewhat different. So the first thing to highlight is that the promoter 5' end versus the 3' end, it was really the 3' end that was giving us much more information than the promoter was. And we didn't get much improvement by combining the two of them together. And as I mentioned, if we only look at the 20% of the genes that really have big expression differences, then we have a really accurate model. If there's real subtle differences between maize and sorghum, we're not able to pick those out particularly well. But if there's a big difference, we can create a classifier that's actually really accurate. But the interesting thing is most of the signal was coming from the terminator end of the sequences. And to plot that out, again, we can, once we've created these models, we can go through base pair by base pair and ask, what did the model learn? What base pairs were most important for making that prediction? And so that's what you're seeing here is essentially these are called saliency maps. And what they're doing is they're telling us what base pair by base pair is providing the most information in high expression. And it turned out when we do that ortholog contrast model, the sequences right before the terminator sequence were the most important for determining the expression levels. While if we look at pseudogenes, 5' UTRs were most important, and so on. So we are learning that for each of these different models, different portions of the gene seem to be most important for explaining this. So what this is suggesting is the model that Hai was working on was really asking questions about how different classes of genes control their regulation. That seems to be an issue that's substantially regulated by the 5' UTR, the promoter sequence, and so on. How the fine-tuning of how one gene changes its expression through evolutionary time seems to be coming more from the 3' UTR side. So whether the gene is on or off, 5', Whether that exact amount of expression that's left on and the fine tuning of it and what makes the difference between maize and sorghum may well be coming from the 3' UTR side of things. So at least that's our current hypothesis. So I want to end up here, though, is can we also probe these models and get down to that level where we're really starting to ask the question, can we edit? And Catherine took one of her models. She lined up her data and her model of knotted one against empirical data that Sarah Hake's group produced a decade ago, and where they tweaked individual base pairs and then did a competition assay to ask, do these individual base pairs matter? And what you'll see is that where she predicts high regulatory probabilities, that's what we see, and where she predicts low in these individual base pair changes, they line up with the competition assays. So I think we've gotten to a level where these machine learning models can be trained, that they have the sensitivity to ask a question if 1 or 2 bases are changed here, that's likely to produce an effect. So what are the limits of our current models? So, you know, those deleterious models I was telling you about, they're good at splitting neutral from functional. And what do I mean by that? Let's say there's 80 million variants out there. I think we're at a stage where, at least for amino acids, I can tell you these are the 2,000 or 3,000 bad amino acids out there. So from one point of view, we separated 3,000 away from 80 million. That seems really good. We're not very good at telling you among those last 3,000, which is the worst one, okay? We're not very good at ranking within those last 3,000. We're also, when we go across this much diversity, we're missing the regulatory landscape. How maize is controlling its expression versus Arabidopsis, things have changed so dramatically that we can't learn all the rules from Arabidopsis on how each individual gene is being expressed in maize. And of course, we have very diverse physiology. So how are we working to get around that? Well, initially, we only had 4 species of the Anthropogoniae. And so what we're doing now is we've just got started on our project that's been funded by the National Science Foundation with my colleagues Toby Kellogg, Matt Hufford, Jeff Rossabara, Adam Siepel, and Chi Sun. And we're going to focus on one clade. And so why do we care about focusing on a clade? Well, I mentioned initially that maize, we've learned a lot from maize, but with 1,000 genomes worth of sequencing, we only capture about 3 million years of evolution. If we were to apply that same 1,000 genomes worth of information across this entire clade from, say, 900 different Anthropogoniae, we would capture 1.5 billion years of evolutionary time, okay? So we've dramatically increased the total amount of time by 300-fold that we're now sampling. And I was telling you how sensitive evolution is at detecting fitness differences, but now we've turbocharged it. Now we have 300 times more information. And the key thing is within this clade, they all share NADP C4 photosynthesis. So they're all the same, roughly in the big scheme of things, the same physiology and similar developmental patterns. So we're not trying to make models that work all the way out to Arabidopsis. We're working with a group of grasses that, to an untrained systematist, many of them all look very similar to one another, okay? But they've adapted all over the planet. So one way to view this is really saturation evolutionary analysis. Every ancestral base pair here has actually been mutated 50 times somewhere coming out here into those 900 different species. So every base pair has been whacked by evolution mutations all over the place. And so we can actually start measuring constraint directly. These species, although they developmentally look very similar, have actually adapted to an incredible range of also environmental adaptations. From frost to very hot temperatures, drought environments to essentially in swamps. And so from across this entire species, we can learn how these species have adapted to many of those diverse environmental adaptations. And so we're working to essentially sample, sequence, analyze all 900 of those species genomes in the next several years. And we really see that evolution and machine learning, we will be bringing those together, where we see that evolution provides us this near infinite level of mutagenesis. It provides us an ability to see fitness effects that are 1,000-fold more powerful than field experiments. And it provides this incredible range of environmental histories. While machine learning is allowing us to scale to genome sizes, learning can be generalized and we can throw virtually anything into a machine learning model. So heterogeneous inputs can go in, and we can still make sense of it. So these are many of the tools we have here, and this is where we're working a lot now, is to figure out what are the most intelligent ways to combine all these together, so we can identify, prioritize those individual SNPs, so that we can go from a place where we have models of how each of the various steps in that central dogma work all the way out to physiology. We can then test those hypotheses based on how they work in standard variation. If they work well, we go ahead and we start editing and selecting away. So I want to end here that a lot of folks in my group have contributed to these ideas. I highlighted many of the people, the postdocs and students who produce a lot of exact data sets I was talking to you about. But my group and all those great collaborations and data sets we've been a product of are really 3 people in the group really help bring everything together. And that's Cinta Vermey, Peter Bradbury, and Sarah Miller. And I'd be happy to take some questions.
So each sample that you're putting into these models seems like a moving target, right? Developmentally speaking, tissue, cells. How do you control for that? The experiments change throughout the life of the plant.
Right. So, I mean, so the question is, how do you change for different developmental time points and everything? So essentially, the way we've been treating it right now, everything is at the same developmental stage, okay? And so the models we've created are for a developmental state, okay, or stage. And so in maize and sorghum, we can still grow those 2 plants side by side one another. We can collect expression data side by side from one another and make sense of them. One of the good things, though, about expression, the data sets are so rich, you can stage one another against, there's so many orthologs. So it is possible to stage models against one another. And just from the empirical data, you can frequently figure out where you are in development, where you are in various stages. So I think long-term, though, the models absolutely need to be generalized through the dynamic, time changes. And that's not something we're interested in, but we have not worked on yet.
Does that become a problem when you're going to other species that may not develop as sorghum or maize does?
I'm sure there'll be situations where the models will not work as well for certain developmental aspects. But I think a lot of them, a lot of the development is, it is timing here or there, stretched out or extended a little bit more, and that produces the phenotypic effect that we're seeing. But some of the basic patterns in regulation are pretty similar. I mean, the fact that a lot of our maize and sorghum models, the fact that we can even train a model on expression that's between maize and sorghum really well, says the rules are pretty much the same. Yeah.
I'm sorry, so I mean, the B4 that you described has a lot of purging rare deleterious alleles, but not really, like it doesn't really involve mining for and spreading and fixing rare beneficial alleles. And I was wondering specifically about biotic stress, where a rare beneficial allele is much, much more valuable than a gene that everyone already has, because that's probably already worthless.
Right. So yeah. So I think the question is about rare advantageous or adaptive alleles. Well, so first off, there's a certain level where we're never going to be able to capture every adaptive mutation that's out there. But most adaptive mutations if they're really truly are adaptive for that population, they will be common there.
Well, but like if you have a biotic stress, like a new one, or a gene which formerly worked really great against soybean cyst nematode 2 years ago is now worthless because of a change in the target.
Right. Yeah. Oh yeah. No. I agree. It's situations where you have very rapidly changing, a red queen situation like biotic stress. I think different approaches really need to be used to figure out what are the useful alleles there. Red queen, I would say, is not. This is not the approach to go find those alleles. Okay, I think there are other approaches that would be far more advantageous for figuring out disease loci than this. But I would say most of the abiotic stresses, they are, yes, they may fluctuate from year to year, but over deep time, all the organisms have seen drought, they've seen flooding, and things like that. So a lot of those alleles are maintained at reasonable frequencies, and over a wide enough panel, you'll be able to find that that allele is adaptive and it's common somewhere. and it isn't truly, it may be rare in your little panel, but if it really is adaptive, and you are sampling as much diversity as, say, the entire Enterpogani, it'll be common in a number of places. Yeah, correct.
So for the transcription factors, transcription factor binding sites, so since the AUC is so high, 0.99, there must be some features that are really indicative of that particular binding site. So between the transcription factor binding sites that showed a high AUC versus those that did not, are there any trends or common features that
I don't know if Catherine is here. The ones that I know is Catherine here? No, okay. The ones that were not very high, anybody else in the lab wants to correct me, feel free. The ones that were not high, generally the ChIP-seq experiments were pretty bad. Okay, so the biggest thing we've seen for the improvement of the quality of the models is replication of numerous ChIP-seq experiments. And when they are replicated and you line up the experiments over and over again, those sites can be modeled really well. So the neat thing also Catherine has done is she's been able to take those models and compare, for these 100 transcription factors, compare what the model looks like against each one for each transcription factor, and the families of transcription factors fall out of it. So suggesting that essentially it really is learning the basic processes by which these TFs are binding. Yeah.
Yeah, as per your presentation, thank you so much. I'm just looking at the way you're saying there's a change across the time due to mutations and other factors. So does it mean the products that we have today, like if they're resistant for diseases or as the same, will also have a change for tomorrow, either to the worse or to the better? And then is there a way of stopping or minimizing these changes across time? And then if you're looking for resistance.
Here, let's I think the question of whether or not to, can we reduce total mutation rate? Higher eukaryotes have, there is actually probably a fairly strong selection to keep down that mutation rate as low as it possibly can. And it's probably about what, at least this type of polymerase biology can pull off. So I don't know that knocking down, selection has tried to do that really hard to make incredibly high fidelity polymerases. And so I'm not convinced that that's an easy target to do, is to reduce the total mutation rate. We can screen DNA sequencing, though, is so cheap, you can essentially screen through things and just throw out any novel mutation that looks kind of bad. So if you're working in a large breeding company, that that is not a crazy idea. That's how you purge things out. Okay, so what was the for the disease? Yeah. Evolution is, I think this kind of goes back to the same type of question, that evolution is not always a great guide for picking out these abiotic red queen type disease issues, okay? That doesn't mean machine learning couldn't be incredibly powerful for predicting what ligands are going to come together and what, if you're going to do that. So I think there's all sorts of black box types of things that you could have machine learning, predicting, how to deal with disease resistance, but it's not using exactly the same evolutionary thing. You may use evolutionary time to generate the diversity, but it'll be kind of a different question.
Along color lines, now that we have essentially the region that is responsible for heterosis, and we have genome engineering technologies, can we fix heterosis in case that been reached in the next decade?
Well, and that's why Pioneer hired Eli Rogers-Melnick. So, whether or not we could fix heterosis in the, so, I think the first question is whether or not we'll be allowed to edit. Okay, so there are those issues, and I think that's, how society will allow that. But then there are going to be, you'll always be able to make a hybrid that is better than whatever inbred you just made. So just because of distributions. So, and there are a few loci across the genome, I have no doubt, that are truly overdominant rather than being the product of a dosage pattern or dominance. type 1. So I think hybrids will always be better. Whether or not, though, it looks right now, a maize hybrid has 2 times the yield of its inbreds or 3 times its yields of its inbred. Whether or not it'll close down to only 50% better or 30% better, and how quickly we can get there, I don't, I think we can get there. I don't know how quickly we'll get there.
So what do you think is the role of hybrids in evolution? So, I mean, I personally like your concept of deleterious mutations accumulating the pericentromeric regions. The only way to break it is by recombination.
Right.
But, the hybrids are a natural system. And so if they are so much better, I mean, I can see over billions of years genome rearranging, so you don't actually have genes in the pericentromeric regions. Are they more accessible to recombination? Because that's, I mean, we see it in the mutant, so that's possible to do.
Yeah, no, and I think this is where, maybe I didn't emphasize this enough, that that pericentromeric increase, we see it, and it's absolutely there of these deleterious mutations, but there are deleterious mutations everywhere, okay? And they're enriched in the pericentromeric regions, but most of the genes are still out at the arms, and they're still having lots of deleterious mutations. Another thing that Eli figured out was there are processes, biased gene conversion is actually maintaining deleterious mutations at high recombination regions of the genome at fairly moderate frequencies. So there are other processes that are out there that are, so deleterious mutations are everywhere. They just happen to be enriched in the pericentromeric region. So you would still, we see heterosis. It's enriched in that chromosome 5, but we see the evidence of heterosis everywhere. So it's not,
So my question is, if you think that hybrids would have higher fitness in natural situations, then you wouldn't expect to see an enrichment of deleterious mutations there. Maybe it's too much philosophical. We can talk later.
I think, because of deleterious mutation, for example, most polyploidy, okay, I think that's extant, is the product of escaping from deleterious mutations, okay? So that's a way to temporarily get away from deleterious mutation buildup. Now, over deep time, so this is where if you look at the total number of polyploids that have been successful in evolutionary time in plants, in deep evolution, it's 50 different events. But at any one time, there's tens of thousands of polyploids that are evolving, and it allows you to short-term escape from deleterious mutations for a few thousand years.
But there's, the polyploids, I'm not sure if I would agree, because the effect of polyploidy is really down the line. It's not immediate. Like immediately, they're really bad.
Yeah, I think some people would argue against it. Yeah. Okay, we can talk later. So, any other questions? Okay, great. Thank you all.
This has been a production of Cornell University, on the web at cornell.edu.